Nearly 10 million people are parasitized by the tapeworm Diphylobothrium latum in the cooler regions of the northern hemisphere. Those who harbor the parasite may develop a blood condition resembling pernicious anemia. This tapeworm requires 3 hosts to complete its life cycle: a fish-eating mammal such as man or the dog, and 2 aquatic animals, a copepod, one of the crustaceans, and a fish, usually a species of pike. The adult worm is located in the small intestine of the mammalian host. In the copepod, a small larval stage develops, while in the fish host is found the larger and final larval stage. The adult Diphylobothrium latum is ivory-colored, attains a length of from 3 to 30 feet in the intestine of man, and may live there for a year or more. This parasite is called the fish tapeworm or the broad tapeworm because of the width of its segments or proglottids. In the intestine of the host, the adult tapeworm may release hundreds of thousands of eggs daily. When infected people defecate in or near bodies of fresh water, or when raw sewage is discharged into these waters, the eggs become a potential health hazard. The egg averages about 70 microns in length and 50 microns in width. At one end of the light brown shell is located a lid, the operculum. And at the opposite end, there is generally seen a knob-like thickening of the shell. The egg is filled with a mass of yolk cells. As development occurs in fresh water, the early embryo stage is visible. Continuing growth, the embryo becomes spherical, almost filling the egg. The embryo begins to show movement, especially of the hooks, which are characteristic of tapeworm embryos. After a week or more of development, the embryo is mature and it hatches. The hatched embryo is known as a chorocydium. Newly hatched chorocydia swim rapidly and have a rather elongated shape. Here the hooks may be seen again. There are 6 hooks with sickle-shaped ends. The movement of the chorocydium in water is caused by the beating of cilia present on the surface of the spherical larva. The action of the cilia may be studied to better advantage in slow motion. Each chorocydium has 2 small flame cells with vibrating cilia. These structures are part of the primitive excretory system. Chorocydia consist of 2 parts: an outer ciliated envelope, the embryophore, and the inner true embryo, or oncosphere, containing the hooks and flame cells. The chorocydium is the only free-living motile stage in the life cycle of Diphylobothrium latum. Actively moving, these organisms may live for several days in cool water, even though they lack a digestive tract. The first aquatic host of Diphylobothrium latum is a copepod. Several species of the genus Diaptomus and a few species of other genera are susceptible to infection. poricidia are ingested by the copepod. In its stomach, the embryophore or outer ciliated envelope is ruptured, releasing the oncosphere. The six-hooked oncosphere becomes active. Pushing against the intestinal wall with the hooks, it breaks through into the blood sinuses of the copepod. Here, the oncospheres round up and become almost motionless. These larvae are now called Procercoid.